Under normal conditions, we have enough glucose to satisfy our needs. However, under some conditions, the body must make glucose. This is necessary following strenuous exercise to replenish the liver and muscle supplies of glycogen. It also occurs during starvation so that the body can maintain adequate blood glucose levels to supply brain cells and red blood cells. Under normal conditions, these two tissues use only glucose for energy. Glucose is produced by the process of gluconeogenesis, the production of glucose from non-carbohydrate starting materials. Gluconeogenesis, an anabolic pathway, occurs primarily in the liver. Lactate, all amino acids except leucine and lysine, and glycerol from fats can all be used to make glucose. However, the amino acids in glycerol are generally used only under starvation conditions. So if you think about what gluconeogenesis means, gluco meaning sugar, neo, and genesis, so it means we're making new glucose. And gluconeogenesis is used by humans and other organisms to help maintain blood glucose levels. And it helps us to avoid hypoglycemia, which is low blood sugar. So what you're going to see is that we're going to use non-carbohydrate starting materials like glycerol, amino acids, lactate, amino acids, and we're going to convert them into glucose. At first glance, gluconeogenesis appears to be simply the reverse of glycolysis, because the intermediates of the two pathways are identical, but this is not the case because steps 1, 3, and 10 of glycolysis are irreversible, and therefore the reverse reactions must be carried out by other enzymes. In step 1 of glycolysis, hexokinase catalyzes the phosphorylation of glucose. In gluconeogenesis, the dephosphorylation of G6P is carried out by the enzyme glucose 6-phosphatase, which is found in the liver but not in the muscle. Similarly, reaction 3 of the phosphorylation of F6P, or fructose 6-phosphate, catalyzed by phosphofructokinase, is irreversible. That step is bypassed in gluconeogenesis by using the enzyme fructose 1,6-bisphosphatase. Finally, the phosphorylation of ADP catalyzed by pyruvate kinase in step 10 of glycolysis cannot be reversed. The conversion of pyruvate to phosphoenolpyruvate actually involves two enzymes and some unusual reactions. The product is the 4-carbon compound oxaloacetate. Then, phosphoenolpyruvate carboxykinase removes the carbon dioxide and adds a phosphoryl group. The donor of the phosphoryl group in this unusual reaction is guanosine triphosphate, or GTP. This is a nucleotide much like ATP, except that the nitrogenous base is guanine. If glycolysis and gluconeogenesis were not regulated in some fashion, the two pathways would occur simultaneously with the disastrous effect that nothing would get done. 3 convenient sites for this regulation are the 3 bypass reactions. Step 3 of glycolysis is catalyzed by the enzyme phosphofructokinase. This enzyme is stimulated by high concentrations of AMP, ADP, and inorganic phosphate, signals that the cell needs energy. When the enzyme is active, glycolysis proceeds. On the other hand, when ATP is plentiful, phosphofructokinase is inhibited and fructose 1,6-bisphosphatase is stimulated. The net result is that in times of energy excess or high concentrations of ATP, gluconeogenesis will occur. The conversion of lactate into glucose is important in mammals. As the muscles work, they produce lactate, which is converted back to glucose in the liver. The glucose is transported into the blood and from there back to the muscle. In the muscle, it can be catabolized to produce ATP, or it can be used to replenish the muscle stores of glycogen. This cyclic process between the liver and skeletal muscle is called the Cori cycle and is shown here. Through this cycle, gluconeogenesis produces enough glucose to restore the depleted muscle glycogen reservoir within 48 hours.